An otherwise normal person has lost enough blood to decrease his hemoglobin concentration from 15 to 12. Which of the following would be expected to decrease? Well, the change in hemoglobin concentration does not affect the amount of oxygen dissolved in plasma. That would be the arterial PO2. So that's going to be unchanged. The arterial hemoglobin saturation is directly linked to the PO2, and if the PO2 is unchanged, then percent saturation is unchanged. That's the oxyhemoglobin dissociation curve. The higher your PO2, the higher your hemoglobin saturation. On the other hand, the arterial oxygen content will change because with less hemoglobin, you have decreased oxygen carrying capacity by hemoglobin. So there's going to be less oxygen bound to hemoglobin. The amount of dissolved oxygen is the same, but that only counts for 1.5% of the oxygen carried by blood. The amount that can be carried by hemoglobin is going to be reduced. And so when you add those two, the amount carried by hemoglobin and dissolved, the total content is going to be reduced. So the correct answer is C. Number 3, when a climber reaches the peak of Mount Everest, all the following would be expected to decrease except, well, at the peak of Mount Everest, the alveolar PO2 is going to be reduced. Okay? So that means that means if the alveolar PO2 is going to be reduced, then the amount of oxygen that enters into the pulmonary capillary blood, the PaO2 is going to be reduced. And if the partial pressure of oxygen in his blood is reduced, then you know that the hemoglobin saturation is going to be reduced. And that's because of the oxyhemoglobin dissociation curve. Okay? So, arterial PO2A would be reduced. Percent saturation D would be reduced. Arterial oxygen content, if you have less oxygen bound to hemoglobin, the saturation is reduced and total oxygen contents can be reduced. The correct answer here is B. Hemoglobin oxygen carrying capacity is unchanged. Capacity is always your hemoglobin times 1.34. Now, while your percent saturation would be lower, the capacity would be unchanged. Number 4, the most important physiologic variable that determines SaO2 would be PaO2. That's the oxyhemoglobin dissociation curve. PaO2 on one axis, SaO2 on the other. Number 5, a patient with a pulse oximetry reading of 82%. Let's look at this one. And a PaO2 of 60. We know that a PaO2 of 60, typically saturation is 90%. Okay. Below 60, we enter into the steep part of the curve. If at a PaO2 of 60, the saturation is instead only 82%, okay. So at 60 instead it's 82%, we know that the curve has shifted to the right. Okay? And so which of these factors will shift the curve to the right? The correct answer is B, hypercapnia. All right, number 6. Based on your understanding of respiratory physiology, venous blood is going to be slightly more, the correct answer is acidic. Why? Well, it's because venous blood picks up CO2. And CO2 reacts with water, forming carbonic acid. That dissociates into hydrogen ions plus bicarbonate. So if there's an increased concentration of hydrogen ions, the blood's going to be more acidic. I dropped your cap a little bit ago. You all see a cap on the floor somewhere? No, that's right. Cap to this thing here. How about I see you? I hate to lose these things because you know when you lose them they dry out real quickly, and this is a good one.
Did it go under skinny?
Yeah, so it's,
What's that?
you're understanding.
Probably right in front of me and I'm just not seeing it.
I think Cat stole it.
All right, let's keep going. A patient has a PaO2 of 90 millimeters of mercury and hemoglobin level of 12 grams per deciliter. Saturation is 95%. Calculate dissolved oxygen content in blood. Well, to calculate dissolved oxygen content, there's a formula. 0.003 milliliters of oxygen per 100 milliliters of blood, or you could say deciliter, times millimeters of mercury. And you multiply that times the PaO2. We've been given that. The PaO2 is 90 millimeters of mercury. And you see, when you do that, these units, millimeters of mercury, are going to cancel out. And you're going to ultimately get 0.27 milliliters of oxygen per 100 milliliters or deciliter of blood. Oxygen carrying capacity for number 8. That's your hemoglobin concentration, 12 grams per deciliter, times your constant 1.34 milliliters of oxygen per gram of hemoglobin. Okay. Gram of hemoglobin is going to cancel out. So your answer is going to be 16.08 milliliters of oxygen per deciliter of blood. Now, to get the actual oxygen content found in hemoglobin, you take this and you multiply it times percent saturation, okay? Which in this case is 95% or 0.95. That's going to get 15.28. And again, it's milliliters of oxygen, deciliter of blood. Okay. And then finally, if you want to get the total oxygen content, you add the amount that's bound to hemoglobin to the amount that's dissolved. And that gives you a number that's close to 15.55. Okay. Next is the type of question. It's very, very typical of what you'd get if you took the MCAT or DAT. Test to get to veterinary school. They give you a little passage and they ask questions about that passage. So fetal hemoglobin, also called hemoglobin F, is the main oxygen transport protein within the fetus. It differs from adult hemoglobin in that it binds to oxygen with greater affinity. Most forms of hemoglobin, including A, S, and F, are tetramers. They consist of 4 globin chains within which there's a heme group. Now, with adult hemoglobin, those globin chains are 2 alpha chains and 2 beta chains. What makes one chain different from another is the amino acid composition and peptide chains. Okay? Now, in fetal hemoglobin, you have 2 alpha chains and 2 gamma chains. So instead of betas, you have gammas. So it says that the gamma subunit is frequently called the fetal hemoglobin subunit. So number 11, based on this passage, which in following can be inferred? A, the oxyhemoglobin association curve for fetal hemoglobin is shifted to the left compared to normal hemoglobin. Let's go back. 60, 90. CO2. If fetal hemoglobin binds to oxygen with greater affinity, then at the same PaO2, you can have a greater percent saturation. Okay. So 90, you might have 95%. So it tells us that the oxyhemoglobin dissociation curve of fetal hemoglobin is shifted to the left because of this greater affinity. B, fetal hemoglobin can bind more oxygen molecules than adult. No, they both contain 4 heme groups. So the amount of oxygen that the hemoglobin is capable of binding is the same. C, the presence of fetal hemoglobin within a fetal bloodstream promotes the fusion of oxygen from the placenta, across the placenta from the mother's bloodstream. That's true. As blood circulates through the placenta, the fetal hemoglobin attracts that oxygen to a greater extent than the maternal hemoglobin. It pulls it away because of the greater affinity. So that's what allows transfer from the maternal bloodstream to the fetal bloodstream, the greater affinity for oxygen. So the correct answer there is D. Number 12. The above passage suggests that fetal hemoglobin and adult hemoglobin have a similar, the correct answer is quaternary structure. Remember, a protein with a quaternary structure is made up of multiple polypeptide chains. The fact that both molecules have 4 polypeptide chains linked together suggests that they have the same quaternary structure. Finally, number 13, the exaggerated Bohr effect, which says to overcome the decreased tendency for oxygen to dissociate, fetal hemoglobin exhibits an increased hematocrit and an exaggerated Bohr effect. The exaggerated Bohr effect refers to A, the acidic pH has a greater effect on oxygen unloading. So even though hemoglobin binds to oxygen more avidly in the presence of hydrogen, it releases that oxygen more avidly as well. Now, notice that the oxyhemoglobin dissociation curve is a sigmoid curve. It's not a linear curve like this. That's because when hemoglobin binds to, say it's 25% saturated, it binds to a single oxygen. That changes the conformation of the entire hemoglobin molecule so that it then binds with even greater affinity to the next oxygen molecule. So once it binds to one, then all of a sudden it binds much more easily to the next one. And the same thing happens to the next one. So as you go from 25 to 50 to 75, all of a sudden this curve starts to move up like this. So that binding to one oxygen molecule increases the affinity for binding to the next oxygen molecule. So it's not a linear relationship, instead it's more sigmoid. All right. You know, in clinical practice, a lot of times we calculate what's called the AA gradient. The AA gradient is the difference between the alveolar PO2, which we can calculate, and the arterial PO2, which we can get from ABGs, blood gases. Okay. We normally, in an otherwise healthy individual, a normal AA gradient is up to 10 millimeters of mercury. Okay. Now, this normal AA gradient can increase slightly as we age. But for all practical purposes, I'll picture an alveolus. The P uppercase O2 should be fairly similar to the P lowercase a O2. Okay? And this is really pulmonary capillary tube, but it's the same thing as the arterial PO2. So we will allow up to a 10 millimeter difference, but anything more than that many times reflects difficulty with external respiration. I'll give you an example. Let's suppose somebody has congestive heart failure. Okay? Well, in congestive heart failure, as the left ventricle, as a pump backs up, instead of pumping like this, it's doing this. Then fluid backs up behind the left ventricle, back into the left atrium, and then ultimately into the pulmonary capillaries. And as pulmonary capillary pressure increases, the fluid gets extravasated into the pulmonary tissue. So now you start to build up fluid within the respiratory membrane, between the alveolus and the pulmonary capillary. In fact, sometimes the fluid then enters into the alveolus. Well, that's going to impair diffusion capacity of oxygen. And so, as a result of that impaired diffusion, you see a larger difference between the alveolar PO2 and the arterial PO2. Does that make sense? So you could calculate a PaO2 of 100 millimeters mercury, but you may only have an arterial PO2 of 50 millimeters mercury. So what's your A gradient here? It's 50 millimeters mercury. It's much higher than is normal. Now, as you successfully treat the congestive heart failure and the pulmonary edema resolves, what do you notice? That your A gradient begins to shrink. Okay. I should also note that CO2 is rarely affected by pulmonary edema. Why? Because CO2 has a much greater solubility constant. It moves much more quickly through the respiratory membrane. It's not, and so much so that that little bit of fluid that's accumulating within the respiratory membrane doesn't affect it. So whereas pulmonary edema impacts your PaO2, it doesn't affect your PCO2 near to the same extent. So what do we do when someone's PaO2 drops down to 50? We give them supplemental oxygen. We may give them a Veni mask with an inspired oxygen